Okay, you can go ahead.
Okay. Are we online?
Yeah.
Are we live now? Okay. Okay, so huge apologies to everyone. I'm not in my office, and internet and like all connections I could use just crashed, and Naveed is now connecting me by phone. Just to introduce Professor Ron Weiss. Ron, I'm really sorry about this technology not cooperating. So it's a real pleasure to have you here as our speaker. Let me just do a brief introduction, although I think most people know you. Professor Weiss obtained his PhD from MIT in 2001, and he was recruited to Princeton right away and held a faculty appointment between 2001 and 2009. At that point, he returned to MIT as a tenured professor in the Department of Biological Engineering and in the Department of Electrical Engineering and Computer Science. He's also the director of the Synthetic Biology Center at MIT and the PI, principal investigator at the MIT Center for Integrative System Synthetic Biology, which is established in 2013 as part of the NIH-funded National Network of Centers for Systems Biology. So Ron Weiss is really one of the pioneers, of the true pioneers of synthetic biology. And he entered this area as a graduate student in 1996 when he was starting to work on his PhD thesis. And it's very interesting that he realized right away that where science is most exciting is at the interface between these two fields, experimental biology and computer science, that were totally distinct at the time. So the first thing he has done was to help set up a wet lab in the electrical engineering and computer science at MIT, which was really an unprecedented effort. He's now directing a laboratory that programs cell behavior by constructing and modeling biochemical and cellular computing systems. He and his laboratory are using computer engineering principles to program cells, to equip them by sensors and by actuators that can be precisely controlled by analog and digital logic circuits. His group was the very first one to construct systemic gene networks with biochemical logic circuits in Escherichia coli and to perform signal processing to detect specific chemical gradients in response to cell-cell communication. A major thrust of his work is in the engineering of the gene networks that can perform specific predetermined functions similar to logic computation. Is also going above the single cell level into the cell aggregates to perform tasks that are really using the cell-cell communication by quorum sensing. His research continues to rely on the integration of biological experiments and software infrastructures. And these studies go into a number of different areas, including cancer biology, systems biology, neurobiology, and tissue engineering. Over the last years, Professor Weiss has had some major publications that are really shaping up and informing the field. He accumulated 25,000 citations with an index 73. But what is really most interesting to see is that among his top 12 papers that are all cited over 500 times each, 2 are in Nature, 1 is in Nature Methods, 1 is in Nature Biotechnology, and 1 is in Science, and 3 are in PNAS. He was recognized by multiple awards of different kinds. As a young investigator, he was one of the top 100 young investigators selected by MIT Technology Review. He was speaking to both the National Academy of Engineering at their Frontiers of Engineering Symposium and the National Academy of Sciences at their Frontiers of Science Symposium. He is a recipient of the Lawrence Keyes Emerson Electric Company Faculty Award at Princeton, and his research in synthetic biology was named by MIT Technology Review as one of the 10 emerging technologies that will change your world. So, Deron, welcome to Tissue Talks. Thanks very much to accepting the invitation, and now you can start sharing your screen.
Great. Thank you. Thank you. I appreciate the invitation, and I look forward to having this interaction with you, telling you a little bit about what we do in synthetic biology, programmable organoids, and neuromorphic computing, and then getting a chance to interact with you. As Gordon mentioned, I have a background actually in computer science, and so as a graduate student over a quarter century ago, I became fascinated with the notion that we might be able to program cells with the same way that we program computers. So when I look at a cell, I view it as a programmable entity, and what I like to do is basically write up biological programs, often instantiated as computer code, where these biological programs basically code from some kind of desired behavior, some kind of, oftentimes some kind of a sense and respond behavior. And so the idea is that we'd like to then convert this type of high-level description of biological behavior, convert it into genetic regulatory networks that actually implement that behavior, encode these networks with RNA or DNA, and then embed this RNA and DNA into the cells, and that essentially allows us to extend or modify the behavior of cells as if they were programmable. Now, cells can actually naturally be viewed as machines, and this is something that's not unique to synthetic biology, but rather this is a view that anyone can take of cells and how they are able to perform sensory functions. They're able to take sensory information and make decisions on that using some kind of an internal controller, and have that controller then make decisions about actuation, altering gene expression, changes in protein or metabolic activity, and secrete things to the microenvironment in which they exist. And so this view is a very convenient view in terms of understanding what the cells are doing, but it's also important to recognize that the controllers that actually are regulating the decisions in cells are incredibly complex and only understood to a limited extent. So what we do in synthetic biology is we try to fix problems that arise in these kind of control mechanisms, and as well as try to create new cellular functionalities by embedding that DNA or RNA into the cells. And what has been effectively the most dominant way by which we think about regulating, by which we think about modifying what these cellular controllers are doing, is this notion of logic circuits. So it's creating situations whereby the DNA or the RNA can implement multi-input functions that are based on logic, such as AND gates, OR gates, NOT gates. And these operations then basically guide when synthetic genes are expressed in our synthetic gene circuits, or when we activate protein activities and so on. And so this notion of programming cells with digital logic, where the nucleic acids are encoding these type of digital logic circuits, is actually rather reasonable. And I think in certain situations, this is quite a useful and effective approach. But as I'm going to argue today as well, there are many other situations where this notion of logic, binary on-off logic, really does not work very well. It really doesn't match the way the underlying regulatory networks apply. And so I'm going to spend the first half of this talk focusing on more multi-input digital logic circuits, and then the second half of the talk on a different metaphor that we're using that is much more analog in nature, that allows us to control cells in interesting ways. In terms of what we do in the lab, we focus on the foundational aspects of synthetic biology, and then we find ways by which we can use this notion of programming to fundamentally change how we approach a variety of different indications, usually health-related applications. So we work quite a bit on cancer immunotherapy, where we design and build multi-input therapeutic agents that can very precisely distinguish between cancer and non-cancer cells. We work on autoimmunity, where it's similar notions that we engineer these cells to be able to detect abnormal situations, for example, abnormal or undesirable inflammation that we want to be able to immunosuppress. But what I'm going to really spend all the time on today is programmable organoids, which is an area that I've been just completely fascinated in ever since I was a graduate student. This notion that we can build genetic circuit that can guide the development of stem cells into interesting three-dimensional structures. So I'm going to really spend all of the talk today on this particular topic of programmable organoids and tell you various things about it. I'm going to start by talking about some introductory mechanisms to be able to create these organoids via engineered symmetry breaking. I'm going to then focus on being able to take these organoids and be able to create cell type specific expression that allows us in an automated way to take these partially mature organoids and actually create more mature organoids in a cell type specific fashion. And then I'm going to conclude by talking about non-binary cell classification via these notion of neuromorphic circuits. And I'm going to argue that these are really important for being able to control developmental processes, especially to control organoid formation. And so what we do with programmable organoids is we start with iPSCs, create genetic circuits that we embed into these iPSCs. We allow these iPSCs to basically aggregate at the bottom of these U-bottom ultra-low adherent wells. They create spheroids, and then we initiate these developmental programs that we embedded in the synthetic gene circuits. And we pursue kind of the quote unquote the obvious applications where we're looking at organ on a chip applications for assessing drug safety, toxicity, and evaluating efficacy of various drugs, as well as ultimate goal of using these organoids for regenerative medicine, where we would implant these organoids into patients that need them. So we would start, here I'm showing you a picture in these agar-well plates where we're starting with day one spheroids. So we will take about 10,000 cells. We would get them to gravitate to the bottom of these wells, and then they form these nice and fairly reproducible spheroids. And the question is, what do we do then? How do we actually guide the development of these iPSC spheroids towards the formation of complex three-dimensional structures? So the Waddington landscape is a really useful mechanism and abstraction that we can think in terms of understanding how we want to control these processes of starting with iPSCs and then embedding synthetic developmental programs that perform a variety of lineage operations, these operations that are able to control the bifurcation and the maturation of these cell types in this lineage tree-like structure. So we'll start with iPSCs, and over time, we want to be able to take them into the spheroids I mentioned, and ultimately these complex three-dimensional organoids. And so the first thing we do is we grow the iPSCs into small spheroids, and then we initiate the developmental program. And the first thing that we have to do is focus on and achieve lineage bifurcation. So we need some kind of a program module that can guide the cell fate to decision to actually bifurcate. And what happens is, as a result of that, we would get multiple lineages. And these lineages, interestingly, continue along these Waddington landscape trajectories, such that we ultimately, if we do this just the right way, we ultimately get development and actually co-development of all the necessary cell types that we'd like to achieve within these organoids. So this, and I'll show you data on how we do this. What essentially almost always, or perhaps always happens, is that at some point these cells stop maturing. They get essentially stuck in a partially mature state. And at that point, we have embedded in the very beginning in these iPSCs additional genetic developmental programs that would automatically sense that the cells got stuck and automatically activate next step differentiation. So this program module 2 will basically determine that particular cells within this co-developing organoids have reached a partially mature state, and in an automated fashion, they will guide these cell types to mature further while not having any direct impact on the rest of the cells within this co-developing organoid. And as a result of that, we're able to obtain automated guided maturation and ultimately generate vascularized multi-cell type organoids. So let me show you some data here. So the one foray into this was to start with iPSCs and be able to have this initial bifurcation into endoderm and actually generate also mesoderm. This is work that was done by Patrick Gee, a former postdoc in the lab, in collaboration with Mo Ibrahim Khani, who is a postdoc with Linda Griffith. And so what we did is we embedded into these iPSCs genetic circuits using lentivirus, and these allowed us to induce expression of GATA6. Now, at the time that we started this, we didn't quite have the fine-grained control that we have now to be able to generate very tight GATA6 expression levels. But rather what happened is due to the multiplicity of infection of these lentivirus constructs harboring GATA6 expression levels in the population, we actually saw a great diversity of GATA6 expression levels. But rather than treating this as a problem, we actually noticed that this was the reason we were successful. And in fact, what happened is this diversity in GATA6 expression levels was critical for the success of this project because it led to symmetry breaking and emergent pattern formation. So we were able to generate from these iPSCs in a very controllable fashion subpopulations of endoderm that were co-developing with mesoderm. At the time, we were also getting ectoderm, but once we moved into three-dimensional spheroids in these wells, we actually were only getting endoderm and mesoderm. So this was nice. This was interesting. But we are actually asking the question, once we push the cells towards co-developing endoderm and mesoderm, what actually happened? And it was really interesting to see that the endoderm, when we gave it the right push, continued to develop towards hepatic lineages. And we ended up getting both cholangiocytes co-developing with hepatocytes, and the mesoderm continued towards hemangioblasts, and we ended up getting endothelial cells, plus we saw hematopoiesis. So at the end of this process, we actually ended up getting these really interesting three-dimensional, highly vascularized liver organoids that were obtained from an initial population of iPSCs, and they were we were able to generate really large three-dimensional organoids, 150,000 cells, starting from 10,000 cells, millimeters in diameter, and actually, in fact, if you let them continue to grow, they were getting to centimeters in diameter, and also ones that, in addition to the integrated vasculature, contain all the liver cell types that are known to exist in an embryonic liver. And so we got that. And the question is, what is the next step? And so I'm going to focus on cell type specific expression. So as I mentioned, we got all the major liver cell types. We are also able to obtain immune cells that are macrophage, the Kupffer cells, seeing some initial indication of bile ducts and so on. As I mentioned, the very large structures that we're getting, centimeters across, and in terms of functionality, we are able to obtain albumin and urea levels in our liver organoids that were consistent with primary hepatocytes, consistent with adult livers. And so that was very exciting to us. But we are asking the question, did we get perfect liver organoids? So we studied the expression levels of ADME genes, 129 ADME genes. These are genes that are involved in drug absorption, distribution, metabolism, and excretion. And what we saw is that most of the ADME genes in our organoid cells were consistent and comparable to what you see with gold standard primary hepatocytes. But we also noticed that several key genes were actually expressed at lower than desired levels. For example, CYP3A4, which belongs to the cytochrome P450 enzyme superfamily, these account for greater than 75% of total metabolism within humans. CYP3A4 levels were lower than desired. And as you can imagine, this will be an important gene to get correct in a liver organoid. So rather than getting upset over this, we said, oh, great, this is an opportunity for synthetic biology to be able to actually address this problem. How can we create circuits that are able to perform the right maturation in an automated fashion? So we start with iPSCs, as I mentioned, in the first step, diversity of GATA6 expression levels, getting mesoderm and endoderm, getting all these cell types that I'm showing you here. But the problem is that the hepatocytes were only partially matured. And so then the question is, can we embed in the iPSCs a genetic circuit or a genetic module that would be able to detect that specific cell types have now become hepatocytes and actually in an automated fashion express hepatocyte maturation factors, but express them only in hepatocytes, because it's important that these hepatocyte maturation factors are not expressed in the other cell types within the organoid, because they could have detrimental effect on the development of these other cells within the organoid. So what we did is we found out that microRNA 121 could serve as a potentially very good biosensor for and a biomarker for hepatocytes within these organoids. It is low in all the other cell types within the organoid, but it is high in the hepatocytes. And so the idea is, can we create a microRNA 122 sensor that when microRNA 122 is high, will automatically activate expression of CYP3A4 and other hepatocyte maturation factors, such as PROX1, ATF5, and so on. And so we turn to our very kind of vast experience with creating microRNA sensors. So we've been able to do that for a variety of different applications, including cancer immunotherapy. How did these microRNA sensors work? So we developed low microRNA sensors, and the idea with low microRNA sensors is rather simple. You place microRNA target sites upstream and downstream of a gene of interest. When the microRNA is low, the output is high, and when the microRNA is high, the output is low. So this is a low microRNA sensor, but what we actually need is a high microRNA sensor. We want to create a situation whereby when microRNA 122 is high, we express the output. And so how do you build a genetic circuit that can do that? So you basically create a situation where the microRNA represses a repressor of the final gene of interest, such that when the microRNA is low, the repressor is high. As a result of that, the gene of interest is low. And when the microRNA is high, that represses the repressor, allowing expression of the gene of interest. So we said, great, this is something that worked for us in other contexts. The problem is, for greater than 5 years, we were trying to get this implemented in iPSCs, and we're failing. What happened was that these genetic circuits that we're embedding in iPSCs were getting silenced quickly in these iPSCs. And this is, in fact, a dirty little secret in synthetic biology, that when you try to build genetic circuits that operate over long durations, they are highly prone to epigenetic silencing. And as a result of that, we were not able to obtain this desired off to on gene expression that should happen in hepatocytes about 1 to 2 weeks into the experiment. So what we ended up doing is we actually turning to another project that was taking place in the lab. This is a project that was headed by Bree and Noreen, former graduate students in the lab, and they created this PERSIST platform. This is a platform where we're using post-transcriptional regulation with these CRISPR proteins called endoribonucleases. And the key here is that we are doing the regulation not at the transcriptional level, but rather have constitutive expression of our genes of interest with post-transcriptional, specifically translational regulation. And what happens is that if you maintain the genes always on, they do not get epigenetically silenced. And so we have genes that are always on, but we are using translational regulation to ensure that the genes of interest are expressed only at the right time under the right conditions. And so we were able to build these translational off switches. So what's the idea here? That we get constitutive expression of a gene of interest. We place an endoribonuclease site upstream of the gene of interest. And when we add the endoribonuclease, we see that we get really nice repression, up to 250-fold repression in the expression of the gene of interest. We can also create on switches. These on switches basically start off with low expression. The reason they're starting off with low expression is that we are placing these RNA degradation sites downstream of the gene of interest. So normally the RNA is degraded, but when we add an endoribonuclease, what happens is that the endoribonuclease basically cleaves off this RNA degradation site. We have a stabilizer element here, and as a result of that, we have the ability to create nice up to 200-fold activation in response to the endoribonuclease. So we said, let's go ahead and actually use these endoribonucleases at the regulating at the translational level to achieve our high microRNA sensor. So the idea is that the microRNA 122 now regulates an endoribonuclease, which regulates CYP3A4. So in stem cells, the microRNA 122 is low, ERN is high, represses CYP3A4, and in hepatocytes, microRNA 122 is high, represses ERN, and that is, as a result of that, that allows expression of CYP3A4 and induction, timely induction of differentiation in these hepatocytes. As a result of that, the PERSIST approach basically overcomes epigenetic silencing, and we get gene expression to go from off to on 2 weeks into the experiment. So we're able to do that. Let me skip these right here. And we're able to get, in fact, really nice timely activation of CYP3A4 in the organoids. We generated 27 different variants of this circuit, and in the best variants, we are indeed able to get really nice increases in the gene expression for CYP3A4, as well as in the metabolic activity. So as we're comparing the initial GATA6 organoid in terms of CYP3A4 metabolic assays, relative to our best optimized organoid variants that have really nice CYP3A4 activity. So we're able to demonstrate this automated multi-step differentiation, leading into automated generation of more mature liver organoids. We're able to recently also demonstrate this. This is work that was recently published by Lei, a former postdoc in the lab, showing this that this genetically encoded multi-step differentiation also works in other contexts, starting with iPSCs, generated mesoderm and endothelial cells. And the idea is that that's our initial state, and this intermediate state is sensed by these multi-step developmental programs, which then activate differentiation into hematopoietic progenitor cells in an automated fashion. Okay, so the idea is to sense endothelial cell state with microRNA 126 now, and automatically have transition to hematopoietic progenitor cells. So this is the system. We induce with DOX ETV2 that allows us to then get mesoderm and then differentiation into endothelial cells. And once that happens, we also have this high microRNA 126 sensor that is able to sense the differentiation to endothelial cells and then activate gene expression. So what I really wanted to focus on here is this transition from day 0. So you're seeing both microscopy and flow cytometry, and you see how for microRNA 26 high sensor, we start off with a low population, as indicated by the red dots, each dot representing a cell. And over 5 days, we see an increase in microRNA 126 high sensor. I'm also showing you below a microRNA 302 high sensor, which shows the opposite transition of microRNA 302 sensor going from high in iPSC to low in differentiated cells. And indeed, if you look at ddPCR, we see the microRNA 126 going up, microRNA 302 going down, and our genetically encoded microRNA high microRNA sensors also going up for microRNA 126 and down for 302. So the thing that I really wanted to focus on here is the fact that this is not a binary digital decision, right? This is a process that is a gradual process that happens over time. And this is something that I really want to focus on for the second half of this talk. So I'm going to skip over the fact that these I won't get into the details, but these circuits were indeed able to generate these induced hematopoietic progenitor cells in an automated fashion, looking at a variety of different markers. But the question here was, we are able to generate these liver organoids. We're able to generate this conditional multi-step gene expression leading to precision control over development and function. And the question was, can we generalize this to other organoids? And can organoid development in general be regarded as a set of discrete cell states and discrete cell state transitions? And so I'm going to focus on non-Boolean cell classification that we're actually using right now for the automated development of pancreatic organoids, using this new notion of neuromorphic circuits in computing in cells. So I mentioned this current programming paradigm that is dominant, the notion of digital logic. So here on the left, I'm showing you a 2-input function that is instantiated, that is demonstrated as a heat map. This is a 2-input function where it's a digital logic function. We have input 1 and input 2. So if both inputs are low, the output is high. If both inputs are high, the output is high. Otherwise, the output is low. So this is a checkerboard, typical type of digital logic function. My point is that oftentimes it is like fitting a square peg into a rounded hole, in the sense of being able to kind of use this digital logic abstraction for programming cells. And oftentimes what we want to do is be able to program these multi-input functions using an analog regime, using this notion where we want to activate things in intermediate levels with intermediate input concentration. So the question is, how can we be able to build circuits that essentially match the complexity of the underlying biology? I'll show you a specific example that's motivating our work with pancreatic organoids. So the idea is to start with the same iPSCs, generate a diversity of GATA6 expression levels. When the cells become endoderm, however, embed an endoderm sensor that is able to push the endoderm cells rather than the default hepatic lineages towards pancreatic lineages. And what we actually need is not a discrete step, but rather we need to be able to express a pulse of PDX1 expression. So the idea is to be able to embed a genetic circuit in there that can detect that a cell is going through an endoderm transition and express a pulse of PDX1 expression. How can we do something like that? We need non-binary decision making. And I'll show you how to do that with a 2-input non-binary function. So the idea is, and this is showing you data from our organoids. So we start with organoids whose 2 marker levels, marker A, marker B. I'm not naming them here, but we found a variety of different ones that I'm going to show you later. So they start both as low in iPSCs. And as the cells differentiate towards endoderm, you see their transition, the green transition there. And as they differentiate towards mesoderm, the cells take a different path. And what we need to be able to do is express PDX1 during this endoderm transition, not before and not after. And so this effectively means that we need this kind of a bandpass circuit detection capability so that it would activate only when marker A levels are intermediate, and also showing you the appropriate marker B levels. So we need some kind of a bandpass detection capability. How can we do that? Really difficult to do that, if not impossible to do that with digital logic. So we turned our attention into neuromorphic circuits. This is work that was done initially theoretically by Andrew, Christian, Wenlong, and also in collaboration with Elisa Franco at UCLA. And so the notion is that artificial neuronal networks are incredibly powerful classifiers. They're able to take complex input conditions and able to perform these analog computations and be able to make very complex decisions. They have been obviously incredibly powerful in software, in electronic hardware. We want to bring them back into the field of biology, which is arguably where they really, really belong. And so how do we build these artificial neuronal networks in living cells? We need to be able to have inputs, we need to be able to have outputs, and importantly, we need to have processing elements. Now, these processing elements in artificial neuronal networks are known as perceptrons. They have inputs, they're connected to multiple inputs, they could be connected to multiple outputs. Importantly, they actually perform a fairly simple computational function. They perform the computational function, which is a weighted summation of the inputs, plus some kind of a nonlinear activation function, which I will show you in the next few slides. So the argument here is that artificial neuronal networks are the appropriate abstraction to use for programming cells in many situations. So I'll show you some work done by Jean, Charles, and Georg recently in the lab. What are the requirements to create these neuromorphic computing capabilities inside cells? So as I mentioned, weighted summation. So we need to be able to have inputs whose values are weighted. We need to be able to add input values. We need to be able to subtract input values if the weights are negative, for example. We need to be able to have this nonlinear function that I mentioned, and we need to be able to compose these nodes into networks. So those are the requirements. If we can achieve these requirements, we have the ability to create artificial neuronal networks in living cells. How can we actually achieve this with biological regulation? So we turn our attention to being able to build these kinds of circuits. So let's start with an input. So we're going to have an input. We have transcription of the input. So this input can be DNA, but this input can be many different things. It could be gene sensors, it could be microRNA sensors, protein sensors, whatever you want that connects to expression of our synthetic genes. We then have transcription, we have the RNA, we have translation. And so it's simple. We get input resulting in transcription translation into an output protein. We can then do addition simply by adding more of that DNA. So as a result of that, we get more RNA, we get more proteins, and we get the ability to add the inputs. So approximating this notion of XA input plus XB input, simple addition. The more complicated one was subtraction. How can we actually implement subtraction? So we turn our attention into these endoribonucleases that I showed you in this talk previously. And so what do? We have expression of these endoribonucleases, and we embed the endoribonuclease sites upstream of the genes of interest. And so what happens is these endoribonucleases then are transcribed, translated, create the proteins. These proteins bind the RNA and actually sequester it. And as a result of that, we're actually able to implement the subtraction operation. So we basically have the ability to have X2 minus X1 implemented in cells. Now let's look at this. Here I'm going to show you the notation that we're using, transcription translation of the endoribonuclease, essentially subtracting away from the other positive input to create these endoribonuclease perceptrons. Here are experimental results. So these are experimental results where we have about 1 million cells. So we have the ability to modulate X1 input levels, modulate X2 input levels, get the whole range of different X1 and X2 input levels. And then we're observing what is the BFP output levels. And so what you see is that as you get more X2 input, you get higher output. And then as you add more X1 input, you get lower output. And so as we look at this in terms of the various effectively data points along this two input function, we see that X2 minus X1 is the thing we're going to graph first. When we look at the various data points, when x2 minus x1 is negative, effectively meaning that x1 is greater than x2, the output is actually 0, right? We cannot go lower than 0 in biology. And then when x2 is greater than x1, we effectively have a subtraction operation. And the nice thing about this gives us the desired nonlinear function of the two inputs. So we basically get max of x2 minus 1, x2 minus x1, or 0, the desired, as I mentioned, the desired nonlinear function. And so here I'm showing you a variety of different endoribonuclease experimental results. So we are able to build a small library of these perceptrons. We can all, and without getting into the details, we're able to also modify by the input weights, by modifying translation rates, again, showing you a variety of experimental results that we can actually do this. So now the inputs can be weighted as well. And so the next question is, can we compose these into actual networks to be able to achieve more sophisticated functions? So we're going to start with a single node. The single node, as I showed you before, has this type of response where the output is high when x2 is high and goes lower as x1 goes lower. So we basically have this region right here. And then we can add another node. Now, the interesting thing about this second node is what we did is we flipped the weights of these inputs. So in the upper node, x2 is positive, x1 is negative. In the lower node, x1 is positive and x2 is negative. And what effectively happens is that we're now adding this other region where x1, when x1 is high, the output is high. When x2 is low, when x2 is high, that subtracts from x1. And we basically get these two regions right here. And experimentally, what we see is we're able to get this dual region type of function. Again, this interesting nonlinear function of two inputs. And so we can take these nodes, we can compose them into networks, and we can even make more sophisticated networks. So we're going to take this dual region network that I just showed you, and we're going to connect it to another node. Now, this other node, we have an input that is always high. So you can think about this two input function whose output is always high, and we're effectively going to subtract this dual region from this always high node, and that actually gives us this bandpass behavior. This is the thing that we were looking for in the very beginning. So we're effectively doing this region algebra operations in these single cells using this endoribonuclease type of control. We can look at this in terms of 3D. I'll skip over that. But we're able to even create fairly complex three-dimensional, three-input functions. In this particular case, this is an implementation of a three-corner function. So we have an X input, we have a Y input, and we have a Z input. And when we look at this three-dimensionally, you see that we have a three-corner, three-input function. So quite sophisticated behaviors. And here's actually examples of other experimental neuromorphic circuits that we've been able to implement. A whole variety of different kinds of analog responses, nonlinear analog responses that can activate at different customizable intermediate values. Now, as we look at this, we would ask the question, basically any input function, theoretically any input function is achievable. But how do we actually go about designing this? It becomes rather difficult to design these manually, to design these by hand. So imagine we want some kind of a complex target function, which is necessary, let's say, for some kind of multi-step organoid differentiation. How do we go about doing this? And so the idea is to actually use machine learning in another context, be able to train our understanding of biology and be able to then have machine learning generate circuit designs for us automatically based on the biology. And so this is something we're calling the biomorphic network architecture. Due to time, I will not get into the details of this. The bottom line here is that using machine learning, we're able to infer the input-output behavior of all of our regulatory elements. This is what I'm showing you here. We're able to then use machine learning to be able to have very good understanding of these behaviors. It learns how these specific cellular processes are actually operating within the cells. So this is experimentally derived. We can then create biomorphic networks from any list of these parts, right? So we have an unbiased understanding of all of these building blocks, and then we are able to then use the biocompiler to be able to guide the design of these complex circuits. So we have taken hundreds of experiments. We have now nearly 1,000 different circuits that we have built and measured. And the cool thing about these is every time we do an experiment, we're able to learn from that experiment, and that using our machine learning platform, and that adds information to our quantitative understanding of each one of the basic building blocks. And so then we can take DNA parts and be able to use the biomorphic model to predict the behavior of these parts when we put them together. But we can also then, and here I'm actually showing you experimental results where we started from being able to modulate the behavior of these parts in many different ways. So these are experimental results. And what we did, again, just for the sake of time, I'm going to not really get into the details of this, but I want to show you that we took these and just with minimal information from the corner experiments, we're able to then have computational predictions that are incredibly consistent with quantitatively with this experimental matrix. So on the left, you're seeing experimental data, and on the right, you're seeing computational predictions from really a small, very small subset of experimental training results. And so now the question is, if we have a desired cell function, can we use our biocompiler to then tell us what specific DNA to use? So for example, here's just a kind of a toy example. Here's a target function that we may want to use. And here's the process of the biocompiler being able to optimize the generation of a new novel neuromorphic circuit that would actually be able to achieve this function. And so we can have a lot of fun with this and actually have the biocompiler generate many different kinds of biomarker pattern classifiers in an automated fashion. Importantly, circling back to PDX1, all of these biomarker classifiers would give us in the pancreatic organoid pulsatile PDX1 expression. This is a thing that we were looking for in the very beginning. So quick summary, I showed you our programmable organoids where we used gene expression heterogeneity to break symmetry and generate these complex three-dimensional structures, vascularized liver organoids. I showed you the PERSIST platform for being able to do automated multi-step differentiation that overcomes epigenetic silencing. And I showed you our ability to now implement artificial neuronal networks in living cells, which I think synergize and make much more sense for many, many biological regulation circuits and programs that we want to be able to develop. So our synthetic biology systems are really becoming increasingly more complex and are, to some extent, are now a better match for the complexity of natural regulatory systems. I mentioned everyone that was involved in this specific project throughout this, various collaborators that I mentioned as well, funding agencies, and I always end my talks by thanking my PhD advisor, Tom Knight, that got me hooked on synthetic biology as a graduate student, and I've been hooked to it ever since. And thank you for your attention as well.
Thank you, Ron. This was really an incredible example of the convergence of disciplines that were far away from each other in the beginning, and how this convergence is really opening entirely new frontiers. Just very quickly, next week, we have Avijit Chaudhuri from Stanford University speaking about the cell migration and morphogenesis in viscoelastic matrices. So Don, we'll need to let you go soon, but maybe one or two questions. The first one comes from Onur Aydin. Thank you for the fascinating talk. So he says, this may be a naive question, but in your system, it takes very carefully engineered synthetic genetic circuits to develop organoids that have mature phenotypes. How does nature do it? So what is missing on one or the other front?
Yeah, so I mean, nature has much more sophisticated circuits than we are able to engineer now. You know, we're getting closer, but we're still very far from the sophistication that nature really has. And so I think it's important for us as synthetic biologists to be humble and to realize that these cells have just amazing capacity to be able to develop. And what we need to do is find out in situations where, you know, they're not quite developing as we like. So, you know, in vitro, in these situations where we have, you know, petri dishes, where we have these ultra low adherent eubiotic wells, that's not a natural situation for the cells. They're not provided with all the appropriate chemical cues, physical cues, mechanical cues that are really necessary for organ development, for embryo development, and so on. They're missing a lot of cues. So the question is, what are the appropriate cues that we can provide to these organoids to drive them towards appropriate maturation? And what I've shown you is our mechanisms for creating these cues using synthetic gene circuits, so they provide the right push at the right place at the right time, you know, in the right context, in the right amplitude. And we can imagine being able to, you know, connect these to other kinds of cues, to mechanical cues, to microfluidic based cues, to, you know, optogenetics. So, you know, it's really a matter of understanding how our synthetic gene circuits can have two-way respectful interactions with the very complex endogenous machinery to get to the goal that we're looking for.
So maybe, Ron, just one more question about the organoids. I mean, I find it really impressive that instead of doing what the field is doing today, which is deriving the individual subpopulations of cells and then combining them, you just do it like in one pass. And I would be curious, like looking like from the very beginning, like endoderm versus mesoderm, and then downstream, how much difference you see in the behaviors when you take iPS cells from different individuals? And maybe more important, how much control, how much fine-tuning you have, and how much you think you need to have in exactly directing the cells to go very well. I mean, do you give them some initial cues and then let them figure out the rest, or you really go step by step from the beginning to the very end of the differentiation and maturation process?
Great question. So what we try to do is a situation where we want the cells to be able to do as much as they can naturally. So we are trying to figure out a situation where are the most important places to provide the cues so that they initiate these complex developmental programs that already exist in the cells. Those programs take over, but so we provide the initial push, we let the cells develop, and then we find out when do they stop developing properly. And that basically creates situations where we need to be able to then embed the second automated differentiation step, the third automated differentiation step. So I would say it's a matter of finding where the cues are needed. And as you mentioned, you know, for us, it's very important to be able to actually try to mimic biology as much as possible, right? So in a biological context, cells co-develop. And so we believe that this co-development, you know, via the important cell-cell interactions can be a really important factor in achieving the right architectures, achieving the right, course of development, achieving the right kind of maturation. So we really are trying to mimic biology as much as we can,
Yeah.
mimic co-development as much as we can.
Okay, thank you so much. I mean, we will ask you, Ron, like other speakers as well, to please answer the remaining questions directly. We will give you contact information so everyone will get a response. Again, thank you so much for this talk, and see you next week.
Yeah, thank you for the invitation.
Sure.
Bye.
Bye.
Ron.